Hi, thank you. Just on a personal note, I'm a neuromuscular neurologist, and throughout my time in clinical practice, I had the privilege of participating in MDA-sponsored clinics. So it's really a pleasure to be here and to be able to talk with you. So I'm going to talk about the regulatory perspective on clinical development of gene therapies. Oh, the pointer is. Oh, this.
This one to go to the next slide. That one to go back.
And
And pointer.
this guy. Oh, okay. All righty. So first, I'd like to go over some regulatory terminology and also the general landscape for gene therapy regulation at FDA, particularly for rare conditions, and then touch on some of the unique characteristics that make regulation of gene therapies different from that of small molecule drugs. And finally, discuss some programs at FDA to help hasten development and review of exceptionally promising gene therapy treatments. So some important regulatory definitions. So human gene therapy is defined for regulatory purposes as a treatment that seeks to modify or manipulate expression of a gene or to alter the biological properties of living cells for therapeutic use. Gene therapy product, that refers to all products that mediate their effect either by transcription or translation of transferred genetic material or by specifically altering host genetic sequences. And under the Orphan Drug Act of 1983, Congress defined a rare disease as a disease or condition that affects fewer than 200,000 people in the United States. So FDA is a large organization. I found that out for the first time when we had a fire drill. But there are staff in over 200 locations around the globe, and as of 2021 at least, there were people in 20 different time zones. So where does review of gene therapy products take place? And so that's in the Office of Therapeutic Products, formerly the Office of Tissues and Advanced Therapies. We're part of the Center for Biologics Evaluation and Research. In addition to clinicians like me, our review teams include staff with expertise in animal testing and in pharmacology and toxicology, as well as experts in chemistry, manufacturing, and controls for gene therapy products. And then we're often assisted by our colleagues in the CBER biostatistics section. And we'll also often draw on help from colleagues elsewhere in FDA. So for example, if a trial involves use of a medical device, then we'll draw on folks from the Center for Devices and Radiologic Health. Now this figure here, it's very exciting. This shows the number of applications for investigational new drugs, INDs, for gene therapy products over almost 35 years. Now, even though the basic concepts for gene therapies were proposed at least as far back as the early 1970s, it took decades and, as you know, enormous effort involving many brave patients and many pioneers in the field to translate those concepts into practical therapies. And so we're at a very exciting time. This is a bit small, but I can tell you that from 2016 till last year, the number of submissions for new gene therapy products has almost tripled, and that's even taking into account the slight dip due to the COVID-19 pandemic. And another very exciting item is that, so this shows the percentage of investigational new drug applications that have been received by what's now the Office of Therapeutic Products, specifically for rare diseases. And in 2022, that was 49%. And that's very, very encouraging, of course, because in the past there were so few treatments for rare disorders. But since many are caused by mutations in a single gene, they're particularly amenable to gene therapy. And here's some historic milestones for gene therapy products. Approval of Kymriah, which was the first cell-based gene therapy. That was in 2017. It's a chimeric antigen receptor T-cell, CAR T-cell product for refractory childhood lymphoblastic B-cell leukemia. And then that same year, Luxturna received approval. It's the first directly administered gene therapy to treat a form of retinal dystrophy causing blindness. And everyone here, of course, is familiar with Zolgensma, approved in 2019, the first systemically administered gene therapy for treatment of spinal muscular atrophy. And this shows BLA approvals from last year. 5 more products were approved for beta thalassemia, for cerebral adrenoleukodystrophy, hemophilia B, a form of bladder cancer, and also a CAR T-cell product approved for multiple myeloma. So what criteria have to be met for a gene therapy to receive approval? So as with other treatments, small molecule drugs and others, by law, approval must be based on substantial evidence of effectiveness and on evidence of safety. The law states that evidence of effectiveness should be obtained from adequate and well-controlled studies. And while certain aspects of product development are feasible for common diseases, that may not always be the case for rare diseases. And FDA regulations provide flexibility for just that kind of situation. And so let's next have a look at that. One consideration for rare diseases is that there's no specific minimum number of patients that's required to establish effectiveness and safety. Instead, that's determined on a case-by-case basis, and it depends on multiple factors, including persuasiveness of the data, the nature of the benefit provided, or the benefit expected if there's a surrogate endpoint that's used, and I'll talk more about that later. The duration of the treatment or of the exposure, the patient population that would be treated after a therapy receives marketing approval, and concerns regarding potential for harm, adverse effects of the treatment. So I want to turn now to clinical development, the process for small molecule drugs that's more familiar, and from there describe what features of the process for gene therapies are different. So looking at the conventional process, so the 3 phases are outlined here. Phase 1 and phase 2 are exploratory. And the phase 1 trial for small molecule drugs typically are performed in a small number of healthy adults. This phase assesses safety and aspects such as tolerability and maximum tolerated dose. And these trials are typically open label. Now, phase 2, the development of those trials are guided in terms of the dosing and monitoring by data from the phase 1 trials. And these trials involve patients, and the trial primarily assesses the magnitude of the effect of the treatment using particular endpoints. And then that data, in turn, will guide the subsequent confirmatory phase 3 trials, which will involve many more patients, gather data both on safety and effectiveness on a much larger scale. Now, because of a number of important differences with regard to gene therapy versus small molecule drugs, this process is modified in some important ways for gene therapy products. So why is the clinical development of gene therapies different? Well, unlike with small molecule drugs, gene therapies often involve products and technologies for which, at this point, there's comparatively little experience in terms of manufacturing, the use, administration, monitoring. In addition, administering these therapies may involve invasive procedures. So, for example, using a catheter and MRI guidance in the operating room to reach a particular region inside the brain. Those procedures may require general anesthesia, so that's an additional risk. Also, while a key benefit of gene therapy is that it may offer a long-term, hopefully permanent treatment, possibly a cure, but any problems with the treatment, conversely, could be more intense or longer lasting than problems that might arise with a small molecule drug. And we also have to consider some additional possible risks, a few of which are here. One is that insertion of the transgene into the genome of the patient, causing mutation in another gene, and also both in the systemic cells as well as germ cells there, which could be potentially transmitted to the recipient's children. Another possibility is that the activity of the transgene could prove difficult to control once it enters cells. Also, the recipient may have an adverse reaction, so, for example, an immune response to the vector carrying the transgene, or possibly to the transgenic protein itself. And so as a result, clinical development for gene therapies has some important differences. And so we tend to think of the development process as consisting of early phase and late phase trials. The early phase trials are exploratory, but for gene therapy, they're designed to obtain data not only on safety, but also preliminary efficacy of the product. And that information, of course, then informs the confirmatory trials. And so there's some aspects that I want to look into in a little bit more detail regarding the study population, the design of the trials, and the investigational treatment and dose exploration. So the study population, of course, is a major consideration. And so as I mentioned, for small molecule drugs, the phase 1 trials will involve healthy adults and are open label. But for gene therapies, there's a possibility of permanent effects on the recipients. So the participants instead in gene therapy early phase trials are patients with the disease being studied. They may be adults, they may be children or infants. And for conditions like neurodegenerative diseases, treatment may have no possibility of benefit in later stages of disease after neurons have died or muscle tissue is atrophied. So the early phase participants in the trial may be patients with less severe disease rather than what would typically be done, which is having participants with more severe disease. And of course, for rare diseases, the population is much smaller, and so the clinical trials then are going to be smaller, and as a result, that may make it more difficult to identify an effect of the product. And so, and that leads into issues of study design. So it may be more difficult to identify an effect, but certainly, and the effect may be small, but if there is a real effect, we want to be able to find it. And so, because of that, we highly recommend that early phase trials, rather than being open label, they instead be designed to generate data that are as clear and as interpretable as possible. And that means randomized controlled trials with masking, blinding, even for the first in-human trial. And while those early phase trials generally aren't statistically powered to show efficacy, having these kinds of design features, again, even at that early stage, even in first in human, can provide much clearer results, which can significantly speed development of the treatment. Some other important considerations regarding the investigational treatment. If it requires an invasive procedure to be delivered, then preparation for the trial has to include training of the surgeons who will be performing that procedure. And that, in turn, often means that the trial can only be conducted at a small number of sites. In addition, because of the possibility of unforeseen risks, both from the product as well as from delivery procedure, early patients are dosed sequentially in a staggered manner with a reasonable time interval between treatment of the patients, so as to limit any harm that may be identified and be able to mitigate that for subsequent recipients. For gene therapy, identifying an effective clinical dose or doses may be difficult to estimate early in development, and there's a number of technical reasons for that. So, for example, how efficiently does the vector transduce the intended cell type? Or when the product is being manufactured, what percent of the viral vector actually contains the transgene versus the percent of those vectors that are empty? And it may turn out that there's a range of biologically active or effective doses. Other possibilities also are that the available doses may be limited initially by manufacturing issues or by the route of administration. And because the immune system typically reacts to the vector and possibly to the transgene, patients are unlikely to be able to receive a second dose of that product, and possibly, if there's cross-reactivity among the viral vectors, to receive a dose of any other gene therapy product for that disease. So getting the dose right is of course, very, very important and can be much more challenging than with small molecule drugs. I want to turn now to safety monitoring for gene therapy clinical trials. And certainly standard clinical tests are a fundamental part of that. But also there may be need for additional testing. So for example, specialized brain imaging or blood tests for particular subsets of lymphocytes. And one important consideration with regard to safety monitoring and adverse events is that all adverse events are considered relevant. So even if the experimental treatment is being used to supplement an existing therapy that may itself have well-characterized side effects. The duration of monitoring is also an important consideration for gene therapies. And that's typically based on medical knowledge of the disease, based on data obtained from preclinical testing in animal models, and experience with similar products, if that exists. And the duration corresponds to the persistence of the effect in general. Now, for gene therapies, that could be permanent. That's the ideal. So long-term monitoring may be necessary. And for therapies that utilize retroviruses or lentiviral vectors, 15-year monitoring is what's required. But importantly, the clinical development can continue during this long-term follow-up period. Now, FDA has a number of ways to assist sponsors who are preparing to conduct clinical trials for gene therapies. And sponsors can save time and resources by talking with us and getting input well before even submitting an IND application. At the earliest stages of product development, so that's when sponsors still working on developing the product and is actually planning the preclinical animal studies, sponsor can request what's called an interact meeting, that used to be called a pre-IND meeting, to discuss plans and to obtain some recommendations on the most efficient ways to proceed. And then when the development program has moved ahead, where the sponsor is beginning to plan clinical trials, sponsors can meet again with FDA to review that progress and to discuss future steps for product development, if there and other related issues, for example, if there's a need for additional preclinical testing, and that's through the pre-IND meeting. And then after submitting an IND, there are various types of formal meetings that sponsors can request, and FDA staff is also available from the various disciplines for informal interaction as well. I want to turn now to the 5 expedited programs that have been established at FDA to speed development and approval of exceptionally promising treatments. Now, all 5, Fast Track, Breakthrough Therapy Designation, Regenerative Medicine Advanced Therapy, or RMAT designation, Priority Review, and Accelerated Approval, are intended for serious or life-threatening conditions, and that's defined as associated with morbidity that has substantial impact on day-to-day functioning. Now, to receive Fast Track designation, data from preclinical or from clinical studies must demonstrate that the product has the potential to address an unmet medical need. And if a product receives Fast Track designation, the sponsor has the opportunity for increased interaction with FDA staff to speed development. And once, based on clinical results, when a BLA, a marketing application, is submitted, it may be considered under rolling review. So that's where sponsor can submit completed sections of the application for review by FDA as they're completed, rather than putting them all together and having to submit them all at once. To receive breakthrough therapy designation, product has to show preliminary clinical evidence of substantial improvement over available therapy. And so here, in addition to the opportunity for frequent meetings and for rolling review, FDA also makes available additional staff resources for intensive guidance on product development and design of clinical trials. And senior managers at FDA become involved early on to further hasten the process. I want to turn next to RMAT. And this program is the newest of the expedited programs. It was established under the 21st Century Cures Act, which was signed into law December 2016. A regenerative medicine therapy is defined as cell therapy, therapeutic tissue engineering product, human cell and tissue product, or any combination product using such therapies or products. And under FDA's interpretation of the statute, gene therapies qualify, including genetically modified cells that lead to a durable modification of cells or tissues. To receive RMAT designation, the product must be intended to treat, modify, reverse, or cure a serious condition. And it also must show preliminary clinical evidence, indicating the potential to address an unmet medical need. An RMAT designation carries all the benefits of breakthrough therapy designation. So here too, the sponsor has the opportunity for frequent meetings, rolling review, intensive guidance on product development, and involvement early on of senior managers at FDA. And in addition, RMAT provides the opportunity for early interaction to discuss potential surrogate or intermediate clinical endpoints to support an application for accelerated approval, which I'll talk about in just a moment. Priority review can be received by a product that shows the potential to be a significant improvement in safety or effectiveness of treatment, prevention, or diagnosis of a serious condition. Now, the standard review for a marketing application can take up to 10 months, but for priority review, FDA commits additional resources in order to complete that review within 6 months. Now, accelerated approval, that's intended for products that have the potential to address an unmet medical need and provide a meaningful advantage over available therapies. Now, traditional approval involves showing a direct demonstration of clinical benefit using a clinical outcome measure that assesses how a patient feels, functions, or survives. For accelerated approval, the product still has to meet the standards of safety and effectiveness, but here the outcome measure used is a surrogate endpoint that is reasonably likely to predict clinical benefit or an intermediate clinical endpoint. So a surrogate endpoint is a marker, so for example, viral load in HIV, or PET scan to detect certain types of cancer cells, that is thought to predict clinical benefit, but is not itself a measure of clinical benefit. And also similarly, an intermediate clinical endpoint, say for example, decreased 12-month relapse rate in multiple sclerosis, is a measure of a therapeutic effect that's considered reasonably likely to predict clinical benefit of a drug in terms of its effect on irreversible morbidity or mortality. And so this is valuable in situations where the disease course is long and it might take a long time, or in an unusually large clinical trial to assess a clinical benefit directly. For products that receive accelerated approval, post-marketing confirmation trials have been required to verify and to characterize that anticipated clinical benefit. To conclude, developing new treatments is very much a team sport, and we encourage collaboration among everyone involved in the process, scientists, clinicians, patients, advocacy groups, as well as industry and regulatory bodies. We also encourage everyone to take advantage of the opportunities for early communication with us at FDA so that we can work with you to provide input on manufacturing, on preclinical testing, and as well as on clinical trials. And under the right circumstances, for rare diseases, a well-designed early phase trial may provide evidence of effectiveness and safety towards a marketing approval. They can really speed that process. And so that's another reason why we very much encourage designing even first-in-human trials for gene therapy products to provide clear and interpretable evidence of effectiveness by including the design elements I mentioned, randomization, double masking or blinding, and placebo or other concurrent controls. And similarly, we also advocate very much that resolving any manufacturing issues as much as possible, as soon as possible, before beginning the first-in-human clinical trial. So that's a bit of the whirlwind tour. There's, for all the topics I've covered, there's a lot of resources available that can fill in a great many details. And in particular, I encourage you to look at the Office of Therapeutic Products, formerly OTAT, OTP Learn webinar series. It has quite a bit of information. And finally, I'd like to thank my colleagues in